This is Mrs. Vance, and today's lesson is about mitosis. Now, before we can start talking about mitosis, we have to talk just a little bit about chromosome. This is the familiar shape of a chromosome to you, okay? And so this represents a chromosome, but actually it really represents two chromosomes. Chromosomes, remember, during the S phase of the cell cycle, the DNA was duplicated or replicated. And so when you see the chromosome structure like this is the tightly coiled structure that you see during cell division only. And this one actually represents two chromosomes. You've got two duplicate chromosomes that are attached together at a place called the centromere. When they're in this configuration, each half of this molecule is called a chromatid, and the chromatids will separate during mitosis, during the cell division, into two separate chromosomes. As long as they're joined together, they're one chromosome. The centromere is the place where they're joined together, but they're really two chromosomes in one in this configuration. They will become two separate chromosomes during cell division. So here's what happens to the chromatin. When you look at a cell that is not actively dividing, you can't see individual chromosomes. All you see is the grainy stuff in the nucleus called chromatin. This is the DNA that's more opened up. It's more loosely coiled, and that makes sense that it should be more loosely coiled, because when the cell is undergoing its normal everyday activities, you have to be able to unzip part of the chromosome and read the code that's written there to do the different jobs that have to be done in the cell. When it's time for cell division, however, it's kind of hard to duplicate all of that and have a big mess of spaghetti in the bowl, as this looks like here. So what happens is the DNA helix winds up tightly around these histone proteins, and then it coils up even more. And when it's time for cell division to occur, it coils up even more tightly and tightly packed together into the familiar condensed chromosome structure. So here we have one chromosome that is really a duplicated chromosome. Each half of it is one chromatid, and the two chromatids are joined together at the centromere. During cell division, this centromere is going to split in half, and the two sister chromatids will now become separate chromosomes and will be separated from each other. So just so you know what a chromosome is, in your body cells that are not actively dividing, you don't have structures that look like this. You just have the chromatin. But if you could see chromosomes, you would just see half of each one rather than the X shape that shows two different chromatids. Now, mitosis is the process of dividing the nucleus, and the mitosis has four phases. The phases are called prophase, metaphase, anaphase, and telophase. Pro means beginning or before, that's the starting phase. Meta means middle. Ana refers to the pulling apart of the chromosomes, and the telo part means the end. And so we're going to talk about what happens in each of these stages one by one. So in prophase, this is an animal cell. In prophase, you see the centrioles have now doubled, and they start to move apart from each other to opposite sides of the nucleus. This happens in animal cells only because they're the only ones that have centrioles. Remember when we talked about the organelles in the cell, we talked about the centrioles being something that was found only in animal cells, and they were involved in organizing the cytoskeleton and involved in organizing cell division. And so then we're going to see exactly what they do during this unit. The nuclear membrane, nuclear envelope, starts to disappear or dissolve. It's reabsorbed by the ER, and you can now see the chromosomes. The nucleolus that is inside the nucleus in most cells will become less obvious and will disappear, and you'll start to see the more tightly coiled chromosomal bodies. Now remember, in plant cells you don't have those centrioles, but you do still have the formation of the microtubule structure called a spindle. That's prophase, the beginning. So the first part of mitosis, you can see the chromosomes. That's prophase. See the chromosomes, and the nuclear membrane disappears. In metaphase, the chromosomes have now migrated to the center of the cell. There are stages kind of in between prophase and metaphase called prometaphase, where the chromosomes are migrating to the center. But in metaphase, they're all lined up in the middle. Meta means middle, okay? And they're all lined up in the middle of the cell. Spindle fibers, now that the spindle has fully formed, are going to attach to the centromeres. And there's going to be one from each pole attached to each centromere. And basically what happens is these microtubules in the spindle begin to have a tug of war to try to pull the chromosomes to one end or the other. And so, when they're pulling equally, then you'll end up with the chromosomes lined up in the center of the cell. We call this the metaphase plate, or the equator of the cell. It's just the middle where the chromosomes are lined up at the middle. In anaphase, the centromeres split, and now that tug of war that was started It's going to pull the chromosomes, the sister chromatids, which are now separate chromosomes, to the opposite poles of the cell. Also, you see that the cell is elongating. This is kind of like what happens if you're in a tug of war with a bunch of people and you're pulling equally on both sides and somebody cuts the rope. Both sides are going to move quickly back the other way. And this is usually the fastest phase of mitosis. Happens pretty quickly. So the centromere splits, the chromatids separate into separate chromosomes, and then they're pulled toward the opposite poles of the cell. Meanwhile, the cell elongates, getting ready for the next part. And the next phase is called telophase. Telophase, the chromosomes start to disperse. They start to kind of unwind from that tightly wound chromosome structure. The nuclear membrane reforms around the nucleus. And at the same time, or immediately following this, the cytoplasm starts to split. That's called cytokinesis, okay? And you'll end up with two daughter cells that have the same number of chromosomes as the original cell. They will be genetically identical to the parent cell that started the whole process. Now, the process of cytokinesis is not really a phase of mitosis. It happens right at the end of telophase or immediately following telophase. And it happens differently in plant cells and animal cells. In animal cells, you have the development of something called a cleavage furrow. Basically, this is kind of a pinching in of the cytoplasm to separate the cytoplasm into two separate cells. It happens because there's some fibers called actin. These are protein fibers that can contract or constrict, and they basically pull past each other, kind of like a drawstring, to pinch the original cell into two daughter cells. In plant cells, you have a cell wall that forms, and so you end up with the formation of a cell plate. The cell plate occurs because there are a series of vesicles that line up there in the middle and kind of fuse together to form the beginnings of the new cell wall, and they have the cell wall material, and they start to form the cell wall in the membrane to separate the cells. To help you remember the stages of mitosis, you can think of some kind of mnemonic phrase that will help. This is one: pat my alligator tenderly. There are lots of different ones you can choose. You don't even have to come up with a sentence if you don't want to, but it might help you to remember what happens in which order. So, pat my alligator tenderly stands for prophase, metaphase, anaphase, and telophase. Now let's look at some photographs compared with some diagrams showing the same thing. These are in your textbook in Chapter 8. Okay, so here we have the G2 phase of interphase. You can, when you look at the picture of the cell, you can see the spindle fibers. That's actually just microtubules that are organized around the centriole, and you can't see any chromosomes in here. You see a diffuse blue area that is the nucleus of the cell. So in the diagram, you can see that we've got the chromatin. It's already been duplicated because this is the G2 phase, so the S phase is passed. So you've got it there. You can't really tell that it's duplicated, but it has been. You can see the nucleolus, the nuclear envelope, and the plasma membrane. Okay, and you see the centriole pair there, but it's getting ready to start. Something's getting ready to start happening. In prophase, when you look at the photograph here, you can actually see the little spaghetti-like strands of DNA that have already started to condense. And so you see here the chromatids, the chromosome consisting of the chromatids. You see the nuclear envelope here, but the chromosomes are definitely in the center of the cell. But then you see the beginning of the mitotic spindle. This is the animal cell, so you see those centrioles separating and forming the spindle fibers in between them. Those are microtubules that will attach to the chromosomes. In prometaphase, this is kind of between prophase and metaphase, the nuclear envelope is dissolving. Sometimes you can still see part of it there. The chromatids are now even more tightly compacted than they were before. They're moving around in the cell. They're attached to spindle fibers, okay, and the spindle fibers are pulling them into position. Sometimes one end of the cell attaches to the spindle, attaches to the chromosome before the other end does, and so it kind of pulls that way. But eventually they'll be attached on both sides, and the spindle is much more organized now. Then we go to the next picture. We can see in metaphase they've gotten all lined up in the middle in the spindle. Each centromere is attached to two spindle fibers, one from each pole. You see they're all lined up here in the photograph. And now in anaphase, the centromere is split, and you see the pulling apart of the daughter chromosomes, or the sister chromatids that are now daughter chromosomes, being pulled to opposite ends of the cell, and the cell itself is elongating. Then in telophase, you see the formation of the new nuclei. You see the chromosomes starting to disperse somewhat. and the nuclear envelope starting to reform, and then you have the cleavage furrow, which is going to separate the two cells into two daughter cells. Remember that the products of mitosis are two daughter cells that are genetically identical to each other and to the original parent cell. So that concludes mitosis and as a part of the M phase of the cell cycle.